Hi Chem 213 students, and welcome to our very last lecture of Chem 213. This is going to be on phospholipids, steroids, and prostaglandins and terpenes, the last four classes of lipids for us to cover. So last time we talked about triglycerides, which are the dietary fats, and we also talked about waxes, which form protective layers protecting from water on plants and on animals like birds. So now we're going to talk about phospholipids, which are the kinds of lipids that are important for creating the cell membranes that exist in animals and plants too. Phospholipids are ester-like derivatives of phosphoric acid, and they're very similar to a triglyceride, although they have a different shape, which makes them biologically useful in a certain way. So these are going to involve a phosphate. So you may recall phosphoric acid here. Here's a Lewis structure, phosphorus making 5 bonds. It has 5 valence electrons as an atom, so this has a 0 formal charge, and it has 3 hydroxyl groups and 1 carbonyl. This is H3PO4, right? Phosphoric acid. But phosphoric acid, besides having OH groups here, could also have OR groups, so they can have esters. So in terms of their usage in the body and biologically, often there are so-called phosphoesters, where the phosphorus is attached to an oxygen, which is attached to some carbon. You would see these when you learn about DNA too. DNA has a carbohydrate phosphate backbone, where it alternates between a carbohydrate and a phosphate group, a carbohydrate group and a phosphate group. So DNA and RNA also have phosphate esters as an important part of their structure. But when it comes to cell membranes, they contain a lot of phospholipids, and most of these phospholipids are phosphoglycerides, meaning that they're like a triglyceride. However, instead of having 3 fatty acid esters, there's only 2 fatty acid esters, and then one of the esters is a phosphate ester. Okay, and so we can see this phosphorus attached to an oxygen, attached to a carbon here, and this can be either protonated, so these 2 oxygens containing a hydrogen, or it can be deprotonated, in which they'd have 3 lone pairs and 1 bond, and they'd have a negative formal charge. And so this is phosphatidic acid. It's the simplest phosphoglyceride. Most phosphoglycerides that are biologically active also have a phosphoester right here with some other group attached, and then we'll have the remaining bonds to oxygen there. So the most abundant phosphoglycerides are phosphoric diesters. Again, the identity of this group right here can change. At biological pHs, phosphoric acid being a decently strong acid, not a strong acid, but not extremely weak either, this will generally be deprotonated at biological pH, and then we'll have some other group hanging on here, which is the R. And this will be a phosphoric diester. Diester meaning ester here, double bond oxygen, OR here, and ester here, double bond oxygen to phosphorus, OR here. Now, there are 2 very common phosphoric diesters. One of these is called cephalins, and these have the group here, which is called ethanolamine. It's called ethanolamine because if you were to hydrolyze it from this phosphoester, you would get ethanol, 2 carbons, and an alcohol with an amine attached on the second carbon. So it's called ethanolamine. And at biological pHs, like in amino acids, the amine is generally protonated. And so we have 4 bonds and no lone pairs on this nitrogen, giving it a positive formal charge. And also common is so-called lecithin, which has this choline group here. Choline is like ethanolamine, except instead of having hydrogen groups here, like see how it has 3 hydrogens here when it's at biological pH protonated. Instead, it has 3 methyl groups. So that's the difference between ethanolamine and choline. And this gives a nitrogen a positive formal charge, again, because it has 4 bonds and no lone pairs. So the reason why these are so common in in biology is because they're important for forming the cell membrane. So they have this fatty part here, and then they have this quite polar part here. In fact, it's not even just polar, it's ionic. So it's extremely, it's like, that's even, more charge distribution or charge difference than polar, right? We have an extremely negative part here, an extremely positive part. And so this part here at the top is quite polar, and it's attracted to water. And then this part here in the green is completely nonpolar over here, and it's not attracted to water at all. So you can imagine in a cell we have water on the outside, we have water on the inside. And so in the cell membrane, the polar parts right here, these point out into the water portions of the cell on the interior or the exterior, and they're represented by these green circles right here. Now these fatty acid chains, these extend into the interior of the membrane and are attracted to one another. And so we get the so-called lipid bilayer. This is the lipid bilayer that composes the cell membrane. This allows for some passage of things through, but most water-soluble things cannot get through except through a protein gate. And so the surface of the cell has many protein gates and proteins embedded into the membrane in order to help with passage through the cell membrane, and also the structural rigidity of the lipid bilayer here. Now, the result of this is that there's a lot of movement that can happen. There's a lot of fluidity to the membrane because we just kind of have this oily part here in between with these chains that can kind of wiggle around and move. However, and so that allows for fluidity of the cell membrane, but there are additional molecules, proteins and steroids that help to make this a little bit more rigid so it doesn't easily fall apart, because although these intermolecular forces are relatively strong, they are somewhat weak. So things can pass through the membrane, but not very efficiently if they're water-soluble things. And the reason why we have a phosphodiester, or rather having two hydrocarbon chains and the phosphoester at the top, is because this leads to a overall shape here that can form a nice, pretty solid membrane wall. If we had only one hydrocarbon chain, the interior of the cell membrane with the fatty parts would be kind of small, and there'd be a lot of gaps in between the chains. If we had if the cell membranes were made out of a triglyceride, we'd have too much fatty acid chains for the polar head, and the polar head here would be too small, and so that would allow gaps for things to get into the lipid bilayer. The shape here with a large polar head from the phosphoester and two hydrocarbon chains makes the head here about the same size as the tails. So this makes kind of a nice way for these phosphoesters to phosphoglycerides to fit together in a way that makes a cohesive structure. And so that said, there is a lot of fluidity to this structure. The attractions between the polar heads are fairly strong, but they're simply intermolecular forces. They can be easily interrupted, and so often there is a need for greater structural rigidity in the membranes. There is a need for that, and so that's taken care of by steroids, specifically cholesterol for the most part. And then there are other functions of steroids as well. And so now we're going to move into steroids. So steroids can function as chemical messengers. They're also an important part of the cell membrane, as I mentioned. Steroids can be recognized by their ABCD ring system, and so all steroid molecules have this in common. They have three cyclohexane rings bonded in this fashion that are joined together, and one cyclopentane ring. And why cyclohexane? Why cyclopentane? Well, this is where we go back to chapter 4. Why does biology choose certain things? So remember, we saw with the carbohydrates that glucose is the molecule of blood sugar, because when it's in the ring form, it can adopt a conformation where all of the hydroxyl groups are in the equatorial position, making for an exceptionally stable cyclic carbohydrate molecule. That's why it is the blood sugar molecule. Likewise, these steroids are made out of cyclohexane and cyclopentane because these are the most stable rings, right? When we get to smaller rings, we get a lot of ring strain. When we get to larger rings, we also get more ring strain, but the ring strain and the steric hindrance is minimized here with cyclohexane, and it is pretty small for cyclopentane as well. And what we get is a series of joined rings. So here we can see them as a series of chair formations. And the result of this is that actually they make quite a flat structure in the end. So we end up with a very flat structure because the steroids are rigid and they have this trans type of connection here. Remember, there is a cis kind of connection where the ring kind of points downward in this direction. But the trans conformation here prevents ring flipping and gives the overall steroid a kind of flat structure to it. And so here's that flat structure. If we're looking at cholesterol, most of this ring here is these joined trans connected here cyclohexane rings that all end up being in this kind of flat conformation. We have the hydroxyl group here on the cholesterol sticking out on the end. We have two methyl groups that are in the axial positions here. And then we have a side chain here. This is this part right here, carbons 22 to 26 and 7 sticking out on the side. And it makes a remarkably flat structure. And as I said, because of this trans joining here, as we discussed way back in chapter 4, this makes it so that a ring flip cannot occur and will be extremely sterically hindered. So cholesterol is important in our body. Our body can synthesize it, and obviously other animals can synthesize it as well. It is synthesized from a terpene called squalene. So I'll show you how to recognize that this is a terpene, but you can kind of see a regularity to its structure, right? Which kind of hints at the fact that it's a polymer. It's a polymer of a terpene. And so that's what we call it, terpenes. But this terpene here can undergo biosynthesis reaction that will be that will produce a cholesterol. Now you can find the complete synthesis in mechanism 26.3 in this chapter. I will not test you on that mechanism, okay? But there is a mechanism provided in the book. What are the steroids hormones used for? As I mentioned, cholesterol is an important part of the cell membranes, but also steroids are used to create the sex hormones. So, contrary to popular belief, you do need cholesterol in your diet. These are used to produce the steroid hormones, the sex hormones. The male hormones are called androgens. Female hormones are estrogens and progestins. So an example of androgens here are testosterone in males. So you can see this is similar to cholesterol. One difference here is the hydroxyl group is instead a ketone, there's no long side chain here as there is in cholesterol. Androsterone here, again, similar. You have the ABCD ring structure, but you just have different functional groups here, the hydroxyl group instead of the ketone. And there's no alkene here for the androsterone. And instead of a ketone group here up at the top, there's a hydroxyl group. You do not need to memorize these names, but you should be able to pick out, what are the functional groups and explain why this is a steroid based on the structure. And so all three types of these hormones are both found in men and women. It's just what differs is the levels of these hormones. So we have estradiol here, and estrone, which are estrogens, two types of estrogens. And so we have a hydroxyl group here. The difference here, this being a benzene ring instead of a cyclohexane in estradiol, and we have a hydroxyl group here up at the top. Notice the positions where we have the most significant differences, right? Between these. In estrone, here the big difference is the hydroxyl group is instead a carbonyl group. But that's the difference between estradiol and estrone. And then progesterone is a progestin. This one has a ketone here. kind of like testosterone does, so it has the same group here. However, unlike testosterone, which has a hydroxyl group up on the D ring, instead there is a ketone group off the ring here. And estrogens and progestins inhibit ovulation during pregnancy. There are also synthetic hormones, which have been designed to mimic estrogens and progestins and inhibit ovulation, and these are used for birth control. Here's two possible types of birth control. Notice they contain the same important functional group, which is the carbon triple bond. So this is one of the few examples of an alkyne in a drug or in a biological molecule. This isn't biological, it's synthetic. But the carbon-carbon triple bond here happens to make this much more biologically potent, and the result is that this can inhibit ovulation and result in no pregnancy, or the inability to become pregnant. So also there's some difference here between ethinyl estradiol and norethindrone. This is but you can see they're both mimicking. The ethinyl estradiol is similar to the estradiol here, and the norethindrone is similar to the progesterone here, in this area here. There's also adrenocortical hormones, cortisol. You've probably heard of that. These are excreted by the adrenal gland, so when you're stressed, this will start pumping out of your body cortisol. Cortisone and cortisol are used therapeutically to prevent inflammation. So this is your body's way of saying, "Oh, we're stressed out. There's inflammation occurring. Let's control that." Okay, and again, you can see the A, B, C, D ring system on each of these. They share this group on the D ring, a carboxylic acid group and a hydroxyl group on the same carbon. They also share this region here on the A ring, which is similar to, as we saw, progesterone here, also similar to testosterone. So cortisone, that ON here, indicating the commonality of these parts of the ABCD ring here. The big difference between cortisone and cortisol is the ketone group here versus the alcohol group here on the C ring. So that's steroid hormones. Now let's talk about prostaglandins, another type of lipid. Prostaglandins are found in all bodies and tissues of fluids. They're generally associated with inflammation, but they are also associated with other functions, including controlling blood pressure, clotting, kidney function, heart function, and so forth. They will contain 20 carbon atoms, and they will have a 5-membered ring with 2 side chains. So that's the key features that you want to look out for when identifying prostaglandins. And these are these 5-membered rings that you may see. They contain a ketone and a alkene group in different positions. So PGA here is a alpha beta unsaturated ketone, alpha, this being the alpha carbon, this being the beta carbon relative to the ketone. PGB, also an alpha beta unsaturated ketone, but it's on the other side here, attached to the larger groups of the prostaglandin. And then PGC here, this is a beta gamma, again being alpha, labeling these here, alpha, beta, this is an alpha carbon, this is a beta carbon. Here this is alpha, beta, gamma. So this is a beta gamma unsaturated ketone. And then there are larger groups on all the prostaglandins. They will differ from one another based on what these R groups are. Here's a few other examples of different prostaglandins here. We can have a beta hydroxy, beta, and so this being the ketone here now on this side of the ring, alpha beta, it's a beta hydroxy ketone. We can have another beta hydroxy ketone, alpha carbon relative to the carbonyl beta, just in the different positions relative to the larger groups here. We can have a diol, and we can have an endoperoxide here. Peroxide meaning endo in the molecule, a peroxide, an oxygen-oxygen double bond. So the PG here indicates that the compounds of prostaglandin, PGA, PGB, PGC are all going to have carbonyl groups and an endocyclic carbon-carbon pi bond. So going back to these, they're all going to have the ketone and they're going to have a alkene group. So that's PGABC. PGD and PGEs are beta hydroxy ketones. So that's these two right here, having a ketone and a hydroxyl group. PGFs are diols, PGGs are endoperoxides. And here's an example of a prostaglandin here. Oops, this is a little my picture here is a little bit messed up. Sorry about that. Just want to make it a little bit bigger. There we go. And also it seems to be cut off a little bit. Sorry about that. But there's the ring over here. I'll show you another example in a second. But this is an example of prostaglandin. The ring's over here. It's hard to see. But it has these two side chains. So let's look at another example. Here we can see this example. So here prostaglandins are going to be formed from arachidonic acid. So this is a polyunsaturated fatty acid, and this can undergo biological reactions to produce the various prostaglandins and another class of molecules called leukotrienes. Prostaglandins belong to a larger class of molecules. They're all called eicosanoids, and they're all derived from arachidonic acid, a polyunsaturated carboxylic acid. Here. But yeah, you can see the two. These are examples of side group here. So this is a PGE prostaglandin. And PGE2 is responsible for muscle contractions during childbirth. The last group of molecules is terpenes. Terpenes are a very, very widely diverse group of compounds. The terpenes are assembled from isoprene. Here, so isoprene is a molecule that it contains 1, 2, 3, 4, 5 carbon atoms, and it has 2 double bonds in it. And these double bonds can undergo polymerization reactions to produce large molecules that are formed from individual units of isoprene, and these molecules are called terpenes. So myrcene is an example of this. So if you have 2 terpene units, let's see. So if you have 2 terpene units, they can join together, and two terpene units right here, and they can form myrcene. And in this bond, this is what was formerly a double bond, and now is a single bond, right? So the isoprene here, I'm trying to get the right orientation here, but you're going to see that these are formed from individual units that can polymerize, and they can polymerize in a variety of ways. The easiest way to see how many units you have, though, is to count the carbons. Each unit has 5 carbons: 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. Okay, and these can polymerize in a variety of ways. Each group is going to have 1, 2, 3, 4, 5 carbons. 1, 2, 3, 4, 5. So this one polymerized with itself. Every time you make a bond, you're going to lose a pi bond. So in these 2 units here, we made 1 bond. So we lost 1 pi bond from the terpenes to produce this sigma bond. Here we have basically 2 bonds because it formed into a ring, and we joined the 2 groups together. And so we lost a couple of double bonds there. Here, this one has 3 terpene units. 1, 2, 3, 4, 5 here. 1, 2, 3, 4, 5, and 1, 2, 3, 4, 5. And this one making 2 bonds, bonding to 2 different groups here, so it loses both of its double bonds. This one bonding to this group, losing one of its double bonds, and this one bonding to this group, losing one of its double bonds. Terpenes are isolated from plant oils. They tend to have nice scents to them. They can be used as flavorants and odorants. So an example of terpene derivatives are menthol, which gives you the peppermint scent, camphor from evergreen trees, and R-carvone, which we used for our polarimetry experiment in Chem 212, also a terpene derivative. In terms of the way terpenes are classified, they're classified based on their number of carbon atoms. So a monoterpene, meaning it comes from 2 terpene units joined together, that has 10 carbon atoms. Sesquiterpene, meaning 3. So this is 2 times 5, right? 2 times 5, so it's 2 units. Is 3 times 5, it's 3 units. Diterpene, 4 times 5, it's going to be 4 units, and so forth. Triterpene, 6 units. 6 times 5. And so we can see another example of a terpene is beta-carotene. Many terpene units, 1, 2, 3, 4, 5, 6, 7, 8. 8 terpene units, so beta-carotene is an example of a tetraterpene. 8 terpene units times 5. 8 isoprene units, rather. And so you can see that a pair of isoprene units is referred to as a terpene, right? So we have 2 pairs of isoprene units, we get a diterpene, 3 pairs of isoprene units, triterpene, and so forth. So beta-carotene is a tetraterpene. Terpenes are biosynthesized from the following compounds, not from isoprene itself, but an analog of isoprene. These contain high-energy phosphate groups attached. So those are what drive the reaction because they're a good leaving group here. And so there are 2 main terpene derivatives like this, that with the 2 phosphate groups, dimethylallyl pyrophosphate and isopentyl pyrophosphate. And what occurs is that there is a cleavage of the phosphoester here, and we get a carbocation here, and then this can go on and react and produce terpenes by reacting with other terpenes here, other isoprene units. So here's the full mechanism. So first we have the hydrolysis, the cleavage here of the bond producing the phosphoester. This can cleave on its own because it's a high energy bond between the oxygen and the phosphorus. And then we have one of these phosphated terpene units is going to go and attack the positive carbon on the group that just had the phosphate leave. And so here we have a source of electrons, which is the pi bond. This is the the electrophile. The nucleophile here, this is the nucleophile. The electrophile here is this positive formal charge on the carbocation. And so then we get a linkage, and this produces for us now this carbon here will only have 3 bonds and no lone pairs. Before it had 4 bonds and no lone pairs, but now it'll have 3 bonds and no lone pairs. So it now bears a positive formal charge. We can get rid of that formal charge by reforming a double bond if we react with a base like an amino acid, the amine group on an amino acid. This will grab a proton and produce a double bond here, and then finally we have a neutral product. But this one can go on and react in the same nucleophilic type way that the previous one reacted, and then we'll add more onto this chain and more and more and more, and this will make us larger and larger terpenes. So in summary, we've discussed the various forms of lipids, including waxes and triglycerides and phospholipids, steroids, also prostaglandins and terpenes. Of those, we had a few important reactions. One was the hydrogenation of oils to produce saturated fats. This is important for the production of margarine. We have the saponification of soap reacting a triglyceride with a base. We have the base catalyzed ester hydrolysis, which produces for us the salts of a carboxylic acid, which is soap. And finally, we had triesterification of triglycerides, which is acid catalyzed to make the carbonyl a better electrophile, and then followed up by a nucleophilic attack by the methanol, and then the loss of the remaining alcohol here, the glycerol, and the production of the methyl esters, which are used for biodiesel. And so that's going to conclude the lipids chapter. And now it's time for you to study for the final exam. I want to wish you the best of luck. And please let me know if you need any help. I'm very happy to help you. Have a great day and good luck on the final.